Thanks for your kind introduction, and I really thanks to President Hae-sung Lee for this wonderful session and for inviting me. So today, actually, I slightly changed my title from Alzheimer's disease to the neurodegenerative disease because I talked Alzheimer's disease, Parkinson's disease, both of the neurodegenerative disease. So title is the reassessing the neurodegenerative disease through sex-specific lens, insight from public and organoid-based transcriptomic data. So actually, last year I already invited this session for the G-STR section. So last year I talked about the sex differences in the progression of glucose metabolic dysfunction in Alzheimer's disease. So in this paper, we found some glucose metabolic dysfunction genes, I mean the female specific genes, and I talked about the insight for the Alzheimer's disease for the precision medicine approach for Alzheimer's disease. So, yeah, sex difference is very important for many neurodegenerative disease. So as you can see here, there are many larger number of women than men have Alzheimer's disease worldwide. Ratio is approximately 2 to 1. So, sorry. So this is the very human-based, publicly available database, and they told the very importance of the sex difference in Alzheimer's disease. So, but actually, I'm an organoid researcher, so I always think that then should sex differences also be considered in vitro model? So that's today's topic. So I'm studying about the brain organoid. Nowadays, many researchers are saying the neural organoid. So let me just briefly introduce what is the brain organoid. So brain organoid and neural organoid is a miniaturized and simplified version of a neural tissue that can be produced in vitro in 3D that shows realistic microanatomy. So I'm studying about the multi-region, the brain organoids and mechanismic studies, and also I'm trying to tracking the many changes and mechanismic changes of the brain organoids in real time. And also I'm currently working on the disease modeling and drug screening and so on. So I published this kind of paper by using the brain organoids. So thankfully, so I get award from the science from the KSBNS, the SciTech Korea Young Neuroscientist Award. So tomorrow I will more focus on the brain organoid and the microglia interaction. So please visit tomorrow again and the Young Scientist Award session. So today, to present the list is importance of reassessing the neurodegenerative disease through a sex-specific lens. And also, I think we need to have a question that do we need to consider the sex difference in vitro model such as brain organoid as well? And third one is I will show you some unpublished data about the organoid-based transcriptomic analysis to reinterpret the sex difference, and I will introduce the platform about the GEOD database and GEO2R analytical tools, and also the take-home messages. So since I'm an organoid researcher, I tried to find the sex difference research by using organoid sample, but it was very difficult to find. Actually, there are no paper about this. So of course, there are some paper about hormone changes of the brain organoid is published in Nature, and there are some review article about using organoids to model sex differences in the human brain, but I couldn't find any paper about the studying the sex differences using the brain organoid sample. So although this is not an Alzheimer's disease paper, but I found on paper which have the very even distribution of female and male organoid type. So there are 6 patients and 6 normal control. So this paper are showing that idiopathic Parkinson's disease. The idiopathic means the cause is unknown. So in this paper, they compared the IPD patient-derived brain organoid, midbrain organoid, versus the normal control midbrain organoid. However, they did not explain about the sex difference for their data. So I tried to reanalyze by using this database. So before we move on, so let me just introduce a quick point. So I think researchers can easily access the publicly available database like this. So nowadays, actually, the sequencing data deposition is practically required for every paper, so making it readily accessible at any time. So I usually access the sequencing data by the GEO database from NCBI. So you can access the GEO database here and type your necessary the data, and you can see the many papers and data here. And also for the bulk RNA sequencing data, so some databases provide us with web-based analytical tools, called the GEO2R. So you can just click the Analyze with GEO2R, and you can just click the Analyze button. Then you can get the many basic transcriptomic graph, like the volcano plot or the UMAP or another quality control data. And also you can get the DEGs very easily. So I just introduced this very good platform. And however, this data set is a single cell RNA sequencing data, so I couldn't do this analysis using the GEO2R. I just download the single cell data and analyzed. So for step 1, so I need to check the organoids can really show the genetic sex-specific genes. So when I compare the female and male organoid sample for both control organoid and IPD organoid, so you can, as you can see here, many the chromosome genes like these yellow genes and the blue genes are brain specific chromosomal gene. So as you can see here, the many genes are significantly different between the female and male patient or the normal control derived organoid sample. So it means that the in vitro 3D organoids also show the sex specific features. Actually, it's quite natural because the authors generate the organoid sample by the patient derived iPSC. So it is quite natural, but I confirm the 3D organoid model also can show the sex-specific features. And for step 2, then which pathways are related to the sex-specific traits? So I got the DGs from the control versus IPD patient for female and male each. So each in the female, there are the many DGs when I compared control versus IPD, and the male also have many DGs between the control and versus IPD. So we performed the gene ontology analysis by using for both the female and male. And very interestingly, the female and male shows the totally different biological process like this. So when I compare the gene ontology data, so as you can see here, the female and male have just only 2 overlap terms. So the 2 overlap terms were modulation of chemical synaptic transmission and the regulation of transsynaptic signaling pathway. There are only 2 pathways overlapped, and other pathways are totally different. So it means that it indicates that IPD related pathways can differ greatly between males and female. So for step 3, then which genes are related to sex-specific traits? So we get the data, the sex-specific gene list in step 1. So these genes. And with these sex-specific genes, it's chromosomal genes, and we put these differentially expressed genes with these sex-specific genes, and we analyze the PPI analysis, the protein-protein interaction analysis, using these genes and the DGs from the control versus IPD, and trying to find the directly related genes with these sex-specific genes for both female and male. So very interestingly, the female and male have different interaction. So for female, when we compare the control versus IPD, so these DGs are, there are many DGs, and this gray area is sex-specific genes. There are two direct interaction between the sex-specific genes and these two genes. So the first one is SMARCA1 genes. It is upregulated in IPD. And second one is EFNB2. It is downregulated in IPD. So SMARCA1 is the chromatin remodeling complex gene, and EFNB2 is the afferent B2 genes. It is very crucial role in the neurodevelopment. On the other hand, in the male, there are one genes are directly linked with the sex-specific genes, the KSBNS. So it is very interact with the neural electrical activity of the brain. So we can check that there are different connectome and different genes are exist between the female and male. So I tried to find what are the genes, and are there any paper about female-specific ID or male-specific IPD traits? But there are no papers. So there are no previous reports the linking these genes and KSBNS to the sex-specific difference in Parkinson's disease. So I think our finding are maybe the very novel observation, and I think this could be an important discovery for understanding the sex-related mechanisms in Parkinson's disease. Actually, this is the unpublished data. So today's talk is unpublished data. So we are currently kept analyzing the sex-specific characteristics of the cell cluster. So since this database is actually single-cell RNA sequencing data, so we can get the UMAP data for the individual single cells. So we are trying to find which cell types are related to sex-specific traits. So we keep trying to understand the IPD sex-specific traits in the future. So this is a summary. So we confirmed that there are clear sex differences exist even in organoid. So this highlights the necessity of considering sex as a factor in vitro model as well. So we also found the IPD-related pathway function quite differently depending on sex. And moreover, we discovered that significant DGs and the directly linked genes in IPD, and these genes vary greatly between male and female. And I think these differences are likely driving the sex-specific phenotypes, and will play a crucial role in advancing precision medicine. So in the future, we will further analyze the cell cluster and diverse data sets using the wide range of public data, such as single-cell RNA sequencing and so on. So I'm really thanks for all the collaborating researchers. And today's data is analyzed by my student Rian Kang. So this is my lab members and this funding source. Thank you very much.